So Hatice, stage is yours.
Thank you for organizers for giving me opportunity to present our work on inferring spatially inferred transcription factor activity networks based on spatial transcriptomics data. Let me give you a brief motivation. As you know, signaling pathways converge on the nucleus, modulate the activity of transcription factors, which cause up and down regulation of their target genes. And these gene expression programs define the phenotype of a cell. This cell signaling is affected by the nearby cells through cell receptor-ligand communication, as well as secreted molecules from nearby cells, such as cytokines. Transcription factors are the main link between this upstream signaling and downstream transcription response, and they are the key modulators of cell fate and function. With the advent of single-cell omics technologies, now we can get a readout of molecular readout from different layers. There are many computational methods developed to infer transcription factor activities utilizing single-cell RNA-seq data, single-cell epigenomic data, and more recently, single-cell multiomics data where we can get both readout from the upstream signaling protein expression and gene expression. These methods didn't take account into how cell identity influenced by nearby cells. But now we have technologies like spatial transcriptomic technology where we can measure both mRNA expression levels across a tissue slide while preserving the global spatial information. For example, one of these technologies are spot-specific technologies. Now it's commercially available from 10x Genomics. Here we are putting the tissues on a slide where we have the barcodes available. When we perform this technology, we get both mRNA level as well as location of the cells. But also, this allows us to get for the same tissue H&E staining, which also giving us the morphological information. One of the caveats with these technologies are they are not single cell resolution yet. These spots contain on the order of 5 to 10 cells. So this is kind of a mixture of cells. But you can get this spatial gene expression. For example, here you are seeing the P3 is expressed in this region and CXCL14 is in this region. With normal single-cell RNA technologies, you are losing the spatial information. By taking advantage of this spatial transcriptomic data, we developed a computational framework for inferring the transcription factor activities called Spatially Informed Transcription Factor Activity Network, STAN. So this is the overview of our methodology. So let me introduce our data types. From the spatial transcriptomic data, we are getting the spot-specific gene expression where we are encoding in this Y matrix. We are also getting the morphological features from H&E staining images as well as the coordinates of each spot where we are encoding in this K matrix, kernel matrix, where we both encode morphological similarity as well as spatial similarity. Additionally, we are using the list of transcription factor curated transcription factor target genes, where we are encoding this D matrix. This is a kind of a noisy prior, but this is giving us a sense of which transcription factor is modulating which gene. Then we are using a regression approach to infer transcription factor activities by integrating spot-specific gene expression as well as spatial features that we obtain from the spatial transcriptomic data. Basically, we are learning this transcription factor activity matrix Y. So this is more biologically inspired dimensional reduction. Now we don't have 20,000 genes, but we are summarizing data in terms of 300 transcription factors. As I mentioned, the data is not single cell resolution, but we can use cell type deconvolution approaches utilizing reference single cell RNA-seq data and infer cell type abundances in each spot. Then, as a downstream analysis, we can use this information to learn cell type specific transcription factors, as well as transcription factors associated with pathological regions. We can also link these transcription factors to upstream cytokine, ligand, and receptor expression. We use publicly available spatial transcriptomic data sets to show the effectiveness of this approach from lymph node as well as from breast cancer patients. And we use HD target database for curating this transcription factor target gene priors. There are many deconvolution approaches, but here we are using for a statistical for inferring the cell type abundances for each spot. As a proof of principle, we first apply our approach to lymph node data, where we know the spatial structure very clearly. In this figure, I'm showing you the clustering of spots based on inferred transcription factor activities. And these are the corresponding spatial locations. You can see that there is kind of clusters are actually having some kind of spatial relationships. For example, interestingly, if you look at cluster number 2, these are overlapping with germinal center annotations from a pathologist. And these germinal centers are specific spatial structures where B cells get mature and proliferate. When we look at the enriched transcription factor activities for germinal center regions, we see the reasonable things, such as batch 2, which has role in maturation of B cells, as well as E2F1, which has role in proliferation function of the cells. Next, we also applied this methodology to breast cancer spatial transcriptomics data. Here I'm giving you one example with triple negative breast cancer. This is the H&E staining and corresponding pathological annotations. The pathologists identified these 6 different regions. We look at what are the enriched transcription factors in these regions. Here I'm summarizing the results in this dot plot. Every circle represents a mean transcription factor activity for that region. Like for example, if you look at FOXA1, you are seeing the high activity in normal stropha and lymphocyte regions, which makes sense because FOXA1 has role for breast development. You can see that activity is higher here. But if you look at the mRNA level, you don't see that association very clearly. As I mentioned, spatial transcriptomic technologies, there is many dropouts, and mRNA levels is going to be very sparse. And if you look at another example, for example, invasive and lymphocytes, you are seeing the higher activity of transcription factors involved in immune pathways, such as IRF2, STAT. You can see that IRF2 activity is high here, but mRNA level, you don't see that distinction. When we apply this to the other breast cancer samples, we find that transcription factors are associated with stroma are very heterogeneous between patients, but transcription factors associated with lymphocytes are common between different patients as well. To sum up, today I show you our computational framework to predict spot-specific transcription factor activities utilizing spatial transcriptomic data with cis-regulatory information. This is more biologically inspired dimensional reduction by predicting the transcription factor activities. We can use this approach to identify transcription factor associated with cell types as well as spatial domains like germinal centers. This work is conducted by former postdoc April Sagan. Now she's a bioinformatics scientist at Grail. I also want to acknowledge our funding resources. Thank you for listening.
Questions? Let me maybe start. So you're using what type of gene transcription factor target gene definitions are you using, and did you check how much this definition has an impact on your observations?
We are using a HD target database, but you can change your transcription factor curated list. And there are different approaches to do this. The best thing is to have the context specific, based on the, for example, epigenomic data, where you do, for example, a motif sequence search, but this is more general. But since we are using this in a regression approach, the model is done weighting the priors which which not helping to explain the gene expression. So by that way, we are like more cleaning this noisy prior.
Last question. Yeah.
Maybe slightly vague question, but what is really what the spatial dimension really adds to this model compared to like what you would be able
For
to do with
example,
single cell? Like, really, is there a quantification of the spatial value?
Yes. Like for example, as I mentioned, this is a regression approach. We are trying to predict the gene expression from these gene transcription factor priors. So when we use the spatial information here, stan, so here every dot represents one spot and the prediction of the gene expression performance between the prediction between actual as well as gene correlation between actual and predicted gene expression. And this is our approach where we use spatial prior. And we also use a baseline approach where we didn't use any spatial prior. You can see that on most of the cases, in the majority of the cases, models with spatial priors performing better than the models without spatial information, as well as the model is performance for improved where spots where we don't have many UMA and mRNA contacts. So that is adding. Another example is for 4 of the patients, we have both spatial transcriptomic as well as single-cell RNA-seq data. And we look at the cell type specific transcription factors using single-cell RNA-seq data as well as spatial transcriptomic data. And we saw that like they are correlated for each cell type.
Okay.
Did I answer your question?
So, yeah.
Thanks a lot.